Texas, Austin. Let's look a little closer at what kinds of information we can get out of sequence comparisons and these phylogenetic relationships. So in this little cartoon, we're just looking at the phylogenetic relationships of the the great apes as well as the ancestors of human beings. Here we're looking at the sequence comparison of cytochrome C. Context: cytochrome C is a critical redox protein in the mitochondrial electron transport chain that supports energy metabolism, and so it's going to be fairly strongly conserved. We see that here 99% sequence identity. That tells us that these proteins are very similar to one another. Even though we're only looking at a few changes in amino acid sequence, though, what we see is very clear relationships that make perfect sense. Homo sapiens are related to chimpanzees in terms of their sequence of cytochromes. That bears out the other observations that we make in terms of the structural relationships between amongst the great apes as compared with other organisms. When we compare Neanderthal, and I mentioned that we are able now to do some sequence comparisons of not exactly fossil remains, but ancient remains, then we can see that there's the sequence relationships between Homo sapiens and Neanderthal are closer than those of either Homo sapiens or Neanderthal with chimpanzee, for example. So powerful modeling that you can do based on these sequence comparisons. And again, for the most part, they support other observations that are based on maybe more gross characteristics. What else can we learn from sequence comparisons? So, heme binding determinants of the globins. We're about to embark in a couple of weeks in a discussion of protein function, and two of the archetypal molecules that we're going to discuss are myoglobin and hemoglobin. I've talked to you already about what myoglobin looks like. It's a globular protein, a bunch of helices arranged in a very complicated structure. What's that complicated structure all about? Well, it's about immobilizing a heme prosthetic group. Those are terms that you need to remember. The heme prosthetic group. The heme actually serves the function of immobilizing an iron atom at its center. Here in this cartoon, you see all these helices packed around this heme. There's the iron atom in the center, right? And there's an oxygen molecule, dioxygen molecule bound at the heme iron. So, in terms of function, what do we expect to be the most strongly conserved aspect of the globin, a globin that binds heme and binds iron? What do you think it's going to be? Well, I hope that you think that it's where the binding actually takes place. So let's look more closely at the binding of the heme. We have the proximal ligand of the iron atom or the heme right here. It's a histidine. I hope you can recognize that that's a histidine side chain. So the histidine forms a strong interaction on the proximal side. This is referred to as the proximal side of the heme with the iron. And on the distal side, we have another heme that's absolutely necessary. Well, I shouldn't say absolutely. We'll see a violation of my absolute there. On the distal side, we see this histidine side chain interacting strongly with the oxygen itself. So mediating that interaction of the oxygen with the globin. So the oxygen is interacting on one side with the iron and on the other side with the distal histidine. So when we look at a sequence conservation, you can look up where I stole this little photograph, this little picture from pandasthumb.org, kind of a nice pro-evolution rant page for those of you that want some of that. Basically, we're looking at a bunch of vertebrate sequences here on the top 6 sequences, and then we're looking at some fish sequences on the bottom. And what we see is in the strongest conservation, I think, is in blue. There's some other color coding going on here. Like we see a strongly conserved proline. I'm betting that that's going to be what? Maybe the break between two helices. Remember, prolines disfavor helices. But when we look at the histidines, right, there's the proximal histidine marked with this asterisk. Strongly conserved, perfectly conserved throughout taxa. Not just looking at warm-blooded vertebrates, but also these cold-blooded vertebrates that I'm showing you down here. If you want to confirm what I'm talking about, you can go look up these boy, I guess these are GenBank accession numbers if you're interested. If we look at the distal histidine on this side, remember, it's less well conserved. Histidine, his, Q, right? So it looks like in some fish, perhaps, we've abandoned the histidine here on the distal side for some other molecule, another side chain, that can serve a similar purpose. But on the proximal side, right, where we're actually determining the binding of the heme to the protein, perfectly conserved histidine. So the point being, of course, oh yeah, here's your legend. The point being here, of course, that we can determine some interesting functions just by gazing at sequences. We don't have to know the structure of myoglobin or of these globins. We don't have to know the protein structure in order to understand the aspects of the sequence that are critical to function. So the assumption here is that sequences, first of all, again, sequences that are similar to each other are between organisms that might be similar to each other, perhaps. Sequences that are similar to each other, though, certainly have structural and functional relationships. That's our assumption. It's borne out when we look at the structures, right? We can see now that there's a reason that this histidine right here is going to be perfectly conserved throughout globin-containing taxa. There's other ways to organize taxa, right? We can do it based on domains. We've talked a lot about domains this week in class. We're going to continue that, but we can also use those structures as a way of organizing organisms as well. Now you'll come back immediately and say, well, doesn't the fold depend on the sequence? And that's true, but there are some compelling reasons for looking at structure as well. I stole this picture from this article here. Oh, I noticed that I have no journal here. This is a PNAS paper. Those of you that don't know about PNAS, that stands for the Proceedings of the National Academy of Sciences, one of the most important journals in all of science. A beautiful cartoon here showing us a tree of life that breaks organisms clearly out into eukaryotes, bacteria, and archaea, and all this done by comparison of domains, right? Domains and structural comparisons. So how do they do it? Well, it starts with the structural comparison of all protein structures, and that's embodied by the SCOP database, Structural Classification of Proteins. This has undergone considerable changes over the years. I think it was first composed in 1994. A lot's happened since then. Now we're looking at 38,000 different protein structures in the PDB, right? That's eliminating a bunch of redundancies, blah, blah, blah. 110,800 domains are used in this analysis, right? That, again, reflecting the complexity of these different protein structures in terms of domains. We can organize those domains into 3,900 families with similar functions, most likely. Those families can be further organized into 1,900 superfamilies. Those can have a variety of different functions, but the same fold, and then yet a smaller number of folds. So 1,195 different folds in the SCOP database. This tells you the repertoire that all living systems have to work with. But when we do this categorization, right, to assemble a phylogeny based on structure, what do we see? Well, in this study, this PNAS study that I cite here at the bottom right, only a few years old, at this time, 174 complete genomes had been worked out, covering all three of these domains of life. Yang et al. Find 49 common structures, right? 49 structures, 49 folds that are observed in all three branches of life. I'm going to let you think about that for a second. 49 structures found in all three branches of life. What's it mean? Well, it means that the common ancestor of all three of those branches had that set, right? It implies, in fact, that there's a common organism, a common ancestor to all three branches of life. And once we start branching out from there, then we see a higher diversity within each subgroup with eukarya versus bacteria, blah, blah, blah. Now, why would we do this? Why would we look at protein structures and try to think about an organization? Well, there are a variety of reasons. Maybe the most compelling one, though, is that structure is more conserved than sequence, right? First of all, the easiest answer there is that there are synonymous mutations at the nucleotide level that don't change protein structure at all. Beyond that, we just showed how there are conservative substitutions, conservative amino acid substitutions that will preserve our function, right? We looked at the globin from the fish that had a slightly different sequence than in terms of heme binding than the warm-blooded vertebrates, right? So the structure is stable to that kind of mutation. So you can assemble a bunch of mutations even at the protein level that don't actually impact the structure, even though they might impact the function, right? So we conserve that structure very much more strongly than sequence. Why is that important? Well, as we're going to see in subsequent slides and subsequent discussions, sequences can diverge very rapidly, especially when we remove selective pressure, right? When there's no selective pressure to maintain a function, then we start to lose sequence similarity very, very rapidly. And so when we try to make these comparisons across time scales, effectively what we're looking at are divergences in time scales of hundreds of millions of years longer, right? Then we start to try to compare sequences that are very, very, very dissimilar. And in fact, something I haven't mentioned is that when we're looking at sequence identity of less than 20% or so, there's no way to draw a clear comparison between those sequences, right? That's just, you've lost all intelligibility in that sequence information. So there's limits to which we cannot go, beyond which we cannot go when we're comparing sequences for trying to make comparisons between those genes. Now, when we look at structures, to come back to this topic, when we look at structures, those structures are going to be more strongly conserved than the sequences that underlie them. So the bottom line here is that we can look back farther in time when we consider structures as our organizational metric instead of sequences. And that can be useful when we're trying to understand divergence of organisms, for example. That's the big evolutionary exclamation mark. But to me, this one is really a beautiful thing, right? All organisms started out with a very small number of available folds, and we radiate and diverge and evolve and develop from there. This is a similar slide to what I showed you earlier when we were looking at those myoglobin sequences. Excuse me. Another little little tidbit on the phylogenetic map. Here we're looking at globin sequence alignments. So leg hemoglobin, this is something that's observed even in some plants, I believe. Myoglobins and hemoglobin alpha and beta, remembering that we're going to come back to the structure and function of the myoglobins and hemoglobin later. But again, these are sequences that we might see in organisms today at year 0, but we can map their sequence divergence based on when we see the emergence of organisms that bear leg hemoglobin and not hemoglobin, for example. Same with myoglobin and hemoglobin. So what we see is that, again, the sequence comparison allows us not only to establish these relationships of organisms, but and most importantly to where we're going to go next, they allow us to map the evolution of sequences. Our fundamental assumption here is about to be that gene duplication leads to evolution of new sequences and new functions. So all organisms encoded the gene for what's now leg hemoglobin, but because we underwent some duplication events, we were able to develop an oxygen storage system, and from that oxygen storage system, further duplications lead to the development of a much more complicated oxygen delivery system. So gene duplication followed by recruitment to new functions That's a complicated idea, so I'm going to spend a couple of slides elaborating upon it. So, what about gene duplication as a driver of evolution? Well, there's a few ideas that I want to introduce that can happen after gene duplication. I'll show you some mechanisms for gene duplication in a minute, but let's just assume that we can duplicate genes. What happens? Well, we can undergo pseudogenization. The creation of pseudogenes. They look like genes, but they don't work. And so we can, the easiest way to envision that is by a mutation induced inactivation of a gene. That mutation doesn't have to change the coding sequence of the protein at all, right? It can merely change the regulatory regions of the protein, for example, and suddenly the protein never is usable anymore because it can't be up or down regulated. But certainly there's other ways to create pseudogenes as well. Deletions, frame shifts, etc. There are duplication events that result in a conservation of function. So in this case, the duplicate genes evolve in concert, or else they are continuously purified. That is, their sequences are maintained as identical or close to identical by a purifying selection, because having duplicates that are identical have some powerful selective advantage. Having twice as much of the gene product, for example, confers a selective advantage, and so you want to maintain that. There is another variety of gene duplication result, that is subfunctionalization. So you can develop a related function from a duplicated gene. We're going to talk a lot about this next week when we think about triphosphate isomerase family of proteins. So easy to imagine, though, if we're talking about a gene that was originally involved in, say, carbohydrate or sugar metabolism, that we could duplicate that gene and then recruit it to a related function in carbohydrate metabolism. That's as opposed to neofunctionalization, which is the development of a completely novel function that's unrelated to the original job that the gene product carried out. So those are 4 classes of result that can happen after gene duplication. But what is gene duplication? So it's a very common phenomenon and is a driver of diversification. 20 to 40% of genes are duplicates across the tree of life. That's an important statistic. Again, reflecting the fact that we start out with a relatively simple ancestor organism that then undergoes a variety of diversifying events. So how do we get these duplications? Well, the Moore article that I gave you to read gives you some nice descriptions of all of those possibilities. These figures that I'm showing you here are stolen from this Trends in Ecology and Evolution article, also fairly recent, nice overview. First of all, we just have a crossing over event, so this could result after or during replication. An unequal crossing over event means we've got, so you can look here, this is one chromosome crossing over with another chromosome at some maybe slightly or more or less homologous region of sequence similarity, but what results from crossing over at this point, right, as opposed to this point, is the creation of a new gene that is neither version 1 nor version 2, but can contain elements of both. I should say too that this isn't necessarily restricted to the transference of one gene. This could be a very large block of DNA containing multiple genes that are duplicated here. And in any case, we get a new structure from this crossing over event. Another one that's very fun to think about in terms of the consequences are this retrotransposition. Excuse me. So, of course, we remember that DNA makes RNA, so we've got our mature RNA. It's got a poly A tail. That's beyond our discussion today. But the key here is that in the mature RNA, the intron, or the unexpressed elements of the gene, right, the non-coding regions of the gene, are spliced out. And so we have a sequence that really does look like the coding sequence of our protein, minus the introns. And so we undergo reverse transcription, and reinsertion into the genome, random insertion into genome. And what happens then is that we have a new gene um, in a different position on the in the genome that lacks all of that complex genetic structure of the parent, right? So mRNA reverse transcribed back into genome sans introns. The article that we read in class also talks about other ways of undergoing gene duplication, slippage of polymerase as the polymerase is transcribing, or perhaps in this case it would be replicating the DNA. Then we jump ahead in the sequence and then add another sequence in. So the slippage of the polymerase on the genome can result in the duplication of large tracts of DNA as well. Likewise, double-strand breaks and can result in the transposition of large blocks of double-strand duplex DNA from one position of the chromosome to another. So there's at least 4 ways of getting at a duplicated set of genes. Now, on the right here, I'm talking about divergent expression. So we're sort of ignoring the coding region of the gene, perhaps, and instead thinking about regulatory regions. So we undergo a duplication event, perfect copies of one another, but here we're describing something that happens, complementary degenerate mutation. So if we think about these maybe being 2 different regulatory sites, right? Let's think of these as cis-acting elements of the DNA, right? Regulatory regions on the DNA. In one copy, the first copy, we have a mutation that results in an altered expression pattern, perhaps, and we have a different mutation that accumulate in the second copy, the duplicated copy of the gene, again resulting in a mutated but different expression pattern. So duplicates require different mutations in regulatory regions. This is going to affect the expression of the gene product in different tissue types, perhaps, or at different developmental times, perhaps, or alters the way that the expression of the gene responds to different stimuli. So that can result in the divergent expression and then ultimately the divergence of the proteins in general. We'll talk more about divergence in class, but I want to turn now to a couple of ideas. This slide, well, I want to talk about divergence and I want to talk about convergence. In this slide, let's look at functional divergence. Here I'm showing you 2 proteins, HSP70, that's a heat shock protein. We're looking 70 again referring to the size of the molecule. This is a 70 kilodalton protein, but what we're looking at is only the ATPase domain, about 30 kilodaltons. Structure, very similar to actin, right? So actin is a structural protein. HSP70 has a very important function that is perhaps rescue of unfolded proteins. It has an enzymatic function, that is, it's an ATPase that hydrolyzes ATP. What I've drawn in these 2 structures, I've color-coded them for you, just choosing a couple of key bits of secondary structure to anchor your eye so that you can see that even though there might be some subtle differences in structure, they are, in terms of their global organization, overall very, very similar. Even though they have, if you look at the sequence comparison here, they have far less than 20% identity. Remember that I told you that about 20% is our bottom limit for being able to align sequences and call them similar. And in this case, we're well below that limit. These molecules are clearly unrelated by sequence, but functionally, but structurally very similar. Functionally different. So we've lost our sequence relationship at a certain level. We've retained our structure, however, but then again we've lost our function. So this gets back to that idea of the SCOP database comparisons, where actually structure can be a better model for looking at relationships than sequence, once those sequential relationships are obscured by accumulation of mutations. Molecules retain sequence and structural relationships. So maybe I was a little overstating the idea here. If you look in green, there are, and it's hard to read, but there are a lot of sequence relationships there. So maybe HSP70 and actin are still, you can still retain some sequence relationships there that are observable. Nevertheless, highly diverged sequences that have been recruited to separate functions that are unrelated. Now, something that's interesting that we're going to come back to in our classroom discussion is the idea of convergence of structures, maybe structures, but certainly functions, as an evolutionary tool. In this case, we're looking at serine proteases. I'll remind you about serine proteases, which we've talked about already a couple of times, and we're going to come back to them again as an enzyme mechanism archetype. The serine proteases are proteases, that is, they hydrolyze peptide bonds, breaking apart proteins. And they do that through the assemblage, in many cases, of this so-called catalytic triad. Here's our serine, hydrogen bonded to a histidine, histidine itself hydrogen bonded to this aspartic acid. So there's an asp-his-ser triad serving to activate this serine as a very strong nucleophile. If we look at the structures of subtlysin on the left, chymotrypsin on the right, here you can see those active sites again, organization of those 3 amino acids identical, totally different structures. No sequence relationship, no structure relationship at all, aside from the way those 3 residues are assembled. So we've got no sequence relationship, therefore we've got no evolutionary relationship, and yet we've developed a similar solution to a chemical problem, that is hydrolysis of a peptide bond. Neat idea. I believe this is my last slide, so I'm going to finish here by saying that most of the time, convergence is very difficult to prove, right? And but there are a lot of people that spend a lot of energy and money trying to do so. So this slide is, this slide is taken from your next reader article. That's an article from Janet Thornton's lab. Tim Barrel evolution. The triose phosphate isomerase is a sort of a canonical structure that I showed you in our protein structure overview a couple of weeks ago. It's a beta barrel, so an alpha beta barrel. We have a parallel beta barrel that's decorated on its exterior by a set of alpha helices. Remember that sort of super secondary structural motif? We assemble that into this tertiary structural of a Tim barrel. Tim barrels are have a very diverse set of functions. There are over 900 different Tim barrels identified in nature. I should check, and we'll talk about in class how many different Tim barrels are encoded in the human genome, right? So, but not all of them are triose phosphate isomerase. So instead, they have been duplicated. That gene has been duplicated and recruited to new functions. Maybe, but maybe what we've converged upon is a similar fold. So I guess the basis of the argument comes from the very diversity of functions, where it sort of becomes a little bit harder to understand how you acquire this new function, this neo-functionalization, that are so distinct from the ancestral Tim barrel. The way we go about doing this, of course, is by looking at the details of the structure. So let's just dive in. And if you want a clearer picture, I hope that you develop a clearer picture when you read the article itself. On this y-axis, we're looking at the number of residues in the Tim barrel protein itself. On this y-axis, we're looking at the number of strands in a particular Tim barrel, and then we're looking at the different families of Tim barrels. So there are a variety of ways of assembling these in their families. Some of them are functional, some of them are just purely structural, some of them are sequence. But in general, there's a consensus that we can assemble the Tim barrels into a finite number of families. And when we look across these, what we see is that, well, there are an awful lot of families that have 8 strands in their barrel, right? The triangles are the strand number, right? The circles are the number of residues. So we see that the number of residues bounces around 300. Compare that back to your Goodsell article where we talk about the size of proteins, monomeric proteins, 300 being our upper limit, so we don't really exceed that very frequently. But then you can see in the error bars in terms of each family, you see there's some variety in terms of the sequence, the size of those proteins overall, but still bouncing around 300. But very rarely do we deviate, right, from 8 residues. Maybe we have 7 strands. Maybe we have 9 strands, but we never have 6 or less strands, and we never have 10 or more strands in our barrels. That's a strong argument from some perspectives that this is these 900 Tim barrels are actually divergent in in uh, in terms of their evolutionary relationships instead of converging on what might just be a very common or easy to achieve structure. Backing up once again, let's go back to me talking about supersecondary structures being assembled into larger protein structures. So you could imagine, from just on the basis of thinking of the beta alpha beta sort of motif as a way of building up larger and larger structures, well, you could imagine then that you might be able to drop a bunch of beta alpha betas together into a gene and create a barrel. This idea of all of the barrels having a similar architecture argues for divergence instead of convergence. And then again, too, we look at the bottom here, we're looking at the number of sequences and versus the number of domains. So the lion's share of Tim barrels exist as single domain proteins. But when we look closer and say that when there are more domains, most of those domains occur either as N-terminal insertions or C-terminal insertion. I'm sorry, N-terminal or C-terminal additions, as opposed to domains here. Let's see. Let's look over at the right. As opposed to insertions. So we can this checker mark here is an insertion. You could imagine that it's easier to envision as a divergence, a structure that involves an inclusion of a new domain at the N-terminus or a new domain at the C-terminus. It's harder to envision how you would drop an insertion of a new domain directly in the middle of your barrel without disrupting the structure and function of that barrel. Not to say that it can't happen, and does happen, but very, very infrequently. So the upshot of this article is that there's a preponderance of evidence that suggests that this very diverse family of 900 Tim barrels actually radiated from perhaps a single ancestor. And there are a variety of lines of evidence that support that. And that's where we're going to wind up next week in class. So read that paper, think about evolution of proteins, and we will discuss sequence conservation and structure conservation and functional diversification next week.